stage where things are conceptually more interesting and challenging. Mathematically, a bit easier. So I will, it's almost philosophical, it's like a non-intuitive. So, let's try that. So far we have talked about that vision is having these 3 stages: encoding, selection, and decoding. Encoding is just take the input and put it in a particular format, okay? Whether it's format of the photoreceptors and the format of the retinal ganglion cell after the processing. And I just want to point out that this efficient coding theory is, you can see it's quite powerful for the retina. And another thing is it actually has an analytical solution. Maybe this was not clear in my teaching, as if all of these things are just drafted by hand. No, it is actually an analytical solution. But in order to help you to digest, I put them like as if I'm drafting it by hand, yeah? And so it is nice to have a principle that you can derive and such that it can be applied to different situations purely because different situations have different probability distributions of the signals, okay? Whether it's spatial, time, whether it's monkeys, birds, the formulation does not say has to be monkey, it has to be space, yeah? So that's wonderful. And then the idea is, can we do this efficient coding so much that you can go to the next step, and then we find out that it doesn't go much further, okay? By V1, you find things no longer efficient. The hardware really expanded. Obviously, is not for the reason of efficiency. And so therefore, it is kind of scientifically reasonable that theories have a domain of applicability. So maybe we have hit its limit. It just cannot apply. So therefore, you need to do the next step to understand. Okay, so the next is selection. What is selection? I also give you a little bit of an overview of our field. So for instance, in the field, when you're a neuroscientist, you can take an introduction to neuroscience. So this is a very latest, one of the latest, because it's 2020 textbook. So of course it talks not just about vision, it talks about memory, it talks about reinforced learning, all kinds of stuff, smell, touch, motion. It has 2 chapters on vision. So you imagine this book maybe have 20 chapters, who knows, okay? And the first chapter focuses exclusively on retina, such as photoreceptors and all these different cells in the retina. And most of the second chapter is on V1. Okay, LGN is just a little route on the way to V1. Okay, and you can say LGN is almost a copy of the retina. I didn't mention about it. And so therefore, introduction to neuroscience, and when they have vision, it's mostly after V1, very little left. Okay, in some textbook, after V1, 0. This textbook at least put about 10 to 15% beyond. And then you wonder why, okay? So as we pointed out, that retina experiment already started in the 1950s, and V1 experiment already starting 10 years later. Then you wonder, what is it that after this 10 years later, 1960s, things are not making progress, okay? And so therefore, usually it is a scientific question you're asking, okay? So why is progress so much slower beyond V1? Because our brain, if you remember, we have 1/3 of our brain for vision, and V1 occupies 1/4 of the part for vision. So if we can spend just 10 years to understand V1, and if V1 is just 1/4 of vision, you imagine another 10 years, you get 50% of vision, and another 10 years, and 40 years, you can solve vision problem. You understand everything, right? Well, since 1960s, 60 years passed, so somehow things got more difficult. Yeah, experimentally. Okay, what is the difficulty? So for example, remember people just stick an electrode onto a neuron, then put things on the screen and see what kind of input can drive the neuron. They can do it very easily for retina, can do it very easily for V1, but for the later stage, they just cannot find things that easily can drive a neuron. So something, this kind of a procedure, to copy this procedure to the higher stage is not working, but you can also say maybe copy the question of the initial stage to the scientific question at a later stage may not be ideal, okay? So therefore, if you think about scientific question, it may not be ideal, okay? And so therefore, we have seen in particular, retina's efficient coding does not apply to V1. Next, let's look into selection. But you can ask the question, why is next selection? How about next is recognition? Yeah? Now, first of all, you get the camera, sample the image, and then I recognize there's a cat in it and dog in it. Why selection? Okay, so first stage, this efficient coding is to compress the data, then send it to CPU, and then CPU recognize what's in the image. Okay, so selection means that the data is actually deleted. Whatever it's not selected is deleted. Okay, so here efficient coding is to compress the data before you send it down. Why do you want to delete? Okay, so therefore, to say the next is selection is actually a framework. Is this framework correct? Now, if we are barking on the wrong trees, it will make our progress difficult. So let's imagine whether this framework is motivated by facts. And so one of the factors, which is counterintuitive, is bottleneck to perception. So the idea is you actually don't see everything. Okay? I kind of demonstrate to you that you are blind to many things. So for example, here's another demonstration. Can you see the difference between these two images? Even though you have efficiently coded it, compressed it, and if you have 100%, if you have seen any difference between them, raise your hand. Oh, very good. More than, yeah, so, but most of you still do not see the huge difference. In one picture, you have airplane, another picture, you do not have airplane. That means we just are literally blind, yeah? So this is because we can only see what we select to see. If you do not select, you don't see it, okay? So how do you select? So for example, you can look at it by your gaze shift. Then try to look at this airplane engine by your gaze shift. Then try to see, is there airplane engine there without shifting your gaze? Difficult, isn't it? Okay, so you have to kind of is the engine present? If you don't shift your gaze, you don't see it. But nevertheless, that part of the retina still has all the photoreceptors sampling the input. Why can't you see it? But this is experimental facts. So whenever you want to do a theory, you have to respect the fact. You cannot pretend vision doesn't have this bottleneck. Okay? So you have to shift your gaze to do it. And so therefore, selection is obviously a big ghost. If you don't address that, vision is like you. This is not something that you can ignore, okay? So it has to be there. But you can also think about, are you sure? What about alternative framework? So in vision community, and one alternative framework is don't look at vision as encoding, selection the bit you actually recognize, selecting it, then recognize it, okay? Decoding is to recognize it. Okay, it's an airplane engine, it's a person, and so on. But alternatives say, well, I'm going to just call it low-level vision, mid-level vision, and high-level vision. Okay, this has been going on in the vision science community for a long time. Now, why is this alternative not a good idea? Can I ask you? Any ideas? Anybody want to raise their hand? Yeah, why is it not a good idea? Say it again?
It may be difficult to model.
Maybe difficult to model. Okay, why do you want to model?
Yeah, like how do I quantify low level, mid level?
Yeah. Maybe difficult to specify exactly what is, doesn't have to model. Okay, so the idea is it's, what is not, you can talk about 3 level, you can be 4 levels or 5 level. This is not precise enough. It's like hand waving. When it's not precise enough, you cannot even say whether it's right or wrong. Yeah? So that's called unfalsifiable framework. Okay? So it's a reason. Because it's kind of, you can call it and say, can you say it's wrong? So it's like a Wolfram Pauli talking about not even wrong. Yeah? Have you heard about this term? Something is not even wrong because you can't say whether it's right or wrong. Yeah? And then it's not scientific. Okay? But often, of course, people may not be thinking that way, but since this is a physics audience, let's keep this as a scientific principle. There are things you should be able to say right or wrong. If something you cannot say, it's not scientific. Okay? Yeah?
Leveling vision, it's not really optimized. Like, I mean, you need to do a bunch of processes a bunch of times in different levels.
But more than just optimization, how can you say it's wrong? You cannot say it's right or wrong, whether optimized or not. It can never be proven wrong. Yeah? Tell me an experiment in which you can do to say this is wrong. Nothing, even when you optimize or not. There's not a single experiment. Yeah? If you do not define what low level, mid level, then you can never, yeah? Vision obviously is many things, and you can divide them into 3. Why not divide it into 2? Why not divide it? Why not divide it into 10? Yeah? And so therefore, it's not even wrong. So that's the idea. Okay, whether optimized or not is a different question. Okay, very good. And so too imprecise, therefore not falsifiable. And then another one is, this is a very well-known vision scientist who says something much more concrete. Therefore, you can tell him he's right or wrong. So he says, vision is primal sketch. It's a kind of primitive sketch of the world first by your 2-dimensional images. But in the end, you want a 3-dimensional view of the world, so somehow has to get there, okay? So there's a 2.5D. Exactly what does that mean, 2.5D? But at least a little bit more concrete, huh? Okay? It would be nice a bit more concrete, okay? A bit more precise. You can say, oh, actually, vision never go to 3D, or vision does go to 3D. So you can say it may be wrong in some part, not. Does it actually go to 2.5D? Well, so it's more precise. And another definition is vision is an inverse problem of image formation. Okay? So what is the problem with image formation? It's a 3D world. You take an eye, take a camera, you take a photo. Okay? And this is a problem called computer graphics. You can just now make all the beautiful images that looks real. Okay, that problem is already solved. But inverse that is that you take the image, trying to infer what is a 3D world. Okay? And so that's a beautiful definition, isn't it? What's wrong with it? Anything wrong with it? It's an inverse function of image formation. This is falsifiable. Why is it falsifiable? You can say, is it really inverse back? Well, we just falsified it, remember? Yeah, we cannot see. So example, you did not reverse back. You could not see it. So therefore, this theory is already falsified. Yeah? Okay? We saw an example that if you indeed invert everything back, you should see the difference between these 2 images immediately. Obviously, you don't. Yeah? And so therefore, it is already falsified. You could also say, so image formation, and it's already falsified. And you probably have also shown in the internet where people do this called, you can't even see the gorilla in our midst. They have a group of people playing basketball with each other, and while they're playing, you're supposed to say how many times the ball is passed from the white T-shirt, because some people white T-shirt, some people black T-shirt. They've passed from the white T-shirt team member 1 to the other. Okay, so you have to count. The ball passed once, twice, so you're just watching that video. And in the middle of the video, there's a gorilla just walk in and wave to you. People do not see it. They are so attended, selecting just to look at the white T-shirt. They did not see it. And so this video can play for a minute. People did not see it. And they say, did you see the gorilla? Half of the people did not see it. And they just could not believe it. So go search in the internet. You'll find that. Yeah. So that's how blind. And the gorilla even go like, say hello to everybody. But actually, it's a gorilla. Somebody in gorilla suit. And so in a sense, you can say that this framework is also falsified, because it does not have this bottleneck to say something you should be blind. And this blindness is too huge for you to ignore. Okay? So therefore, the only framework that with a selection in there, this is the first one. Okay? It doesn't mean that maybe 10 years later we find it's wrong too, but at least this is so far the best we have. Okay? So now let's go to selection. And so it really is centers on selection, because if you don't select, you don't even see it. So therefore, it put it in a central role. And before 2014, there's ongoing debate the attentional selection, whether it's early or late. Now, what does that mean by early and late? To say, oh, I did not see the gorilla, I did not see the airplane engine, I could not say I see it, but perhaps my brain actually processed it. It's just before it comes to my tongue, I could not, yeah? So does it go all the way you process and you recognize the airplane engine, and when you ask for it, you just could not say. So this is called late selection. That means you actually process the airplane all the way. It's just the last step you did not complete, okay? Then this is called late selection. Early selection is that you did not even process it, okay? So let's say you did not even process it in the sense you did not even recognize this. It's not just your memory did not remember. And that's early. And so people have been debating early or late in psychology and just do not have a sure conclusion. Now the consensus is perhaps a mixture of early or late, a mixture of early and late, okay? So early selection, for instance, these people talk about is visual inputs are only processed according to image components such as luminance, color, edge, and never get into detail about, oh, it's an airplane, nothing, okay, not even a surface. And the late selection is object identification is performed in parallel over the whole visual field before you select, and which only subsequently limits which of these identified objects is selected to influence your cognitive report, say, did you see it or not see it, okay? And so all of this debate and going on, so therefore, we put this in the middle. We are almost trying to say it selects very early, yeah? We are almost biased to say it's early. Especially, you are probably thinking, I'm trying to say it selects at V1, because V1, the efficient coding is hitting the wall, maybe selection right there, okay? That's only a guess. Just because you guessed it, it may not be wrong. It may not be correct, okay? But we can guess. And once you select, maybe you initially select by looking at it. So shifting your gaze to it is one way of selecting. And then we say, oh, the airplane engine, let me look a bit more. So there may be a FIFO or feedback loop there, okay? So therefore, the idea is you need to select it before you decode it, okay? This is the early selection. You did not even proceed further, yeah? Okay. So how do you select, okay? So imagine an animal in a world. They need to select things that's most useful to them for survival, whether it's a predator, food. You have to say, I need to go hunt the food. If I do not select the food, I don't get it, yeah? Or if I do not see a predator, I'll be eaten. So you better notice these things that are important to you. But if you, but let me go to a bit more detail. Early selection. Does it start in V1? So in a sense that these psychological debate, by not connecting with neurophysiology, it's difficult, become very unconcrete. Now, if you can connect with neurophysiology, maybe we can make progress, okay? So that's why I say today it's getting a lot of a theoretical question, formulation, philosophical, okay? So therefore, then you say, why was it that you tell me that you cannot see? How come I don't feel it? Yeah, I think I see everything clearly. Until you show me the airplane I did not see, And so this is the difficulty in theoretical formulation. People in early framework, they never put the selection as even a part of it. And so therefore you can say that theoretically we are blind to the scientific question. So why was it that attentional selection was absent for so long in previous framework English? Why is that? That's quite philosophical, yeah? Why didn't people notice it? Yeah? Even though they have been debating whether it's early selection or late selection in the psychology community, but the vision science community, they should know there's a selection and put it in here. Why didn't they put it in there? So that means we have not been appreciating this is an important problem that should be in the center of the framework. Okay, so let's have a thought experiment. Imagine you're born blind, yeah? Your name is Mana? Yeah. Okay, imagine you're born blind, yeah? Let's say you're born blind. Would you know you're blind? You would never know until your parent tells you, oh, you're blind. What do you mean I'm blind? I'm okay, yeah? Say it again?
When I stand in front of mirror, so I can look.
You are standing in front of a mirror. Really? But if you never experienced what do you mean by seeing? Then how would you know you're blind? Does that make sense? if you don't know what seeing is, let's say somebody tells you that by a sixth sense they sense something, you do not understand what that means. Yeah? So this is very difficult. So you may even abstractly accept the fact that your parent tell you you're blind, but standing in front of a mirror, how would you know what to see? You are blind. Does that make sense? Yeah? Okay.
Aren't we like biologically wired to want to sense, like to experience certain things?
Yeah.
I mean, we see the animals that
Yeah.
instinctively seems like they
Okay.
instinctively seek things out.
Yeah. Okay, imagine you cannot sense magnetic field, right? Did you know? You did not know, right? You did not feel you're blind. But some birds can sense. So therefore, this is a very philosophically difficult thing to appreciate, yeah, in a sense that how many people of you think, it's actually, it does make sense. If my parent never tell me I'm blind, or nobody else tell me I'm blind, I will not know I'm blind. How many people think this way? Okay, maybe about half, yeah? So therefore, it's not very easy. And so, it's a hard thing to expect. For instance, if you describe to people, oh, I smell banana. Let's say another person could not smell banana. And another person say, what do you mean banana? Tell me you smell banana, yeah? Can you describe to this person what does it mean by smell banana? It is very difficult to say. Bats can use sonar and the bat say, well, I see that insect with my ear. What do you mean you see that insect by your ear, yeah? So it is very difficult to tell. Okay, so therefore, we did not know we were blind because your parents are as blind as you are. Let's imagine that all of these comes in as 100% of information, and you only select 1%. So 99% is deleted. But your parents also have 99% deleted. So your parents could not tell you you're blind. They did not think they were blind, so they did not think you're blind. No? So therefore, in our human species, we are all as blind as another person. So none of us know we're blind. And so if the scientist, you have to define what vision is. If you did not know you're blind, then you do not put this blindness into the framework of vision. Does that make sense? So this is the challenge that we're facing because we did not know. It's like it's unknown. If you did not know, you did not see the airplane engine. You did not know airplane engine was there, so you did not think you're blind. No? So that's the idea, okay?
Maybe it's an unrelated question. When we talk about hallucinations, which part?
It's not in here. Hallucination is a bit too much. It's in the decoding bit, okay? So yeah. Because hallucination, you're already recognizing something. Even if it's fake, you're recognizing it, okay? So here is still selecting. And the idea is, there's also a distance between disciplines in biology, engineering, what does that mean? There's lots of problems, okay? So in the sense that this is, attentional selection is often studied in the psychology community, and these are studied in computer vision or biology community. So not talking makes it difficult to define a problem, appreciate a problem. Okay, so let's, attentional selection is an unknown, therefore it's difficult. It's like if we did not know there's dark matter existing, will we know the dark matter there? The gravity makes us realize there must be some dark matter, but what if it's not even gravity can make us sense that we will not know, yeah? So that's the idea, okay? We are blind to our blindness. That's a problem. So that makes studying vision a very challenging problem. And another thing is there's a problem with words. Psychologists like to talk about attention. What does that mean by attention? Your teacher tells you, okay, children, pay attention. You're in primary school, please look at me. So this sounds a bit soft science, but actually it's selection of data, okay? So sometimes you need to kind of be a bit precise, just like you need to define what you mean before you start talking about. Otherwise, people say you're hand waving. And if you're hand waving, that also impedes your progress in scientific research, okay? So intuitively, attention is associated with voluntary attention that, I want to listen to the teacher, therefore I voluntarily look at the teacher. And it's conscious and it's a higher level thing. But actually, there's lots of, a lot of selection is actually involuntary. Why? So for example, if it's all voluntary, I will see all the beautiful things in the world. I never see any ugly and evil things because I don't like to look at them, no? So you can decide not to select them, right? Yeah, then everything in the world is beautiful. Nothing evil about it. So if it's all voluntary. Now, because if such a case, the tiger jumps at me, I don't see it, I'll be eaten. So I better have to have some involuntary make me see the tiger and run away. Okay, you cannot only see the good things. However, when you talk about attention, people always associate with, so therefore this is often people not pay attention. This is a big problem you have to do. You have to feature in your theory, okay? So selection, a lot of it is involuntary. And you have to select before decoding. So let me give you, and so therefore recognizing and asking the right question is critical, okay? So therefore, I am going to tell you a little bit about attention, scientific facts, okay? Some facts because you need the data before you can, appreciate the problem. And so let's look at the brain, and the camera receives the input. And the input have 1 million pixels. How much input is that? It's 1 megabytes of data per image. How much is 1 megabytes of data per image? And you also have 30 images per second, right? 1 megabyte of data is a huge book of text. So therefore, each second you are having 30 books coming into your retina. Would you have time to read it in 1 second? No. Right? Fortunately, we have efficient coding. You say, hey, it's not exactly 30 books. Maybe I can compress it into not 30 megabytes. But it turns out that efficient coding in the retina does compress it to 1 megabyte. Okay, but 1 megabyte is still one whole book. Can you read it in 1 second? No. How much can you read? 1 page? 1 second? Not even 1 page. Okay, so we are really blind. We read 1 sentence or 2 sentences per second. Okay, so therefore this is 1 megabytes per second. But what we actually read, now you all know the information theory, you can actually calculate. Remember I say, I would like to invite you for dinner. That's more or less 1 second. That's as much as you read, huh? Okay, so therefore we actually only read 40 bits. So therefore, more than 99% blind. But we live in this world so smart, yeah? Despite our blindness, we survive, yeah? Just like despite our blindness to the magnetic sense of the world, we live okay. Yeah? And despite our vision, we are blind to 99%, we are okay. Okay? So this is how much a bottleneck is. That's why you don't see the gorilla. I make you look at for a few seconds, not just 1 second. Okay, then the question is, how do you find this particular sentence in a huge book? They better be important sentence because the sentence has to tell you, oh, this is a tiger, oh, this is my great food, and this is my mother. Yeah? Because these are important sentences for your survival. Don't look at a sentence that's irrelevant. So how do you choose which sentence is important to your survival before you even read the book? You say, oh, I read through a book and find the most important sentence. You don't have time now to read a book. That is a problem. You don't have time to read a book. Then what? So it's like you don't have a chicken, you want to have an egg. And that is a very difficult thing. So somehow the brain has to work out and always choose the right sentence that will ensure your survival. Okay? So this is quite, yeah, that's the idea. And then selection has to be done before the decoding. That means the selecting which sentence has to be done before you read the whole book and see which sentence is important. Illogical, isn't it? Very illogical. So this illogical situation makes theoretical formulation of this problem really, really difficult. Yeah? But nevertheless, that's the fun of studying the brain, because in studying the brain, we encounter these very novel problems. Even though they're challenging, they kind of force us to think about it. Okay? And then you may ask, how did you know it was 40 bits? Okay? I told you can run sentence, one is rough 40 bits. But turns out this was measured in 1950s. How do they measure it? Okay? They have people look at these 16 symbols. Okay? This symbol, that symbol, and say, okay, remember all these 16 symbols. Okay, this person say, and I'm going to show you one picture that will only have one of these symbols on it. Okay, could be this one, and you will just have to tell me which one I just showed you. Okay? So 16 symbols to say which one, how much information is that? 4 bits. Yeah, we now learn the information theory, it's 4 bits. Because 2 to the 4 is 16. Okay? So you have to pick out of these 16 possibility 1, and that's 4 bits of information. Uncertainty removed, okay? And so therefore, you can say, how much time do I have to show you each image before you get 4 bits? You can measure it, okay? So it turns out that if you flash a single symbol in the picture to people for only 1/15th of a second, and show it to 20 observers, and only 1 guessed it correctly. Okay? So of course, if you did not see it, you just randomly guess. If you randomly guess out of 16, you'll have 1/16th chance to guess it just right. So therefore, the fact that only 1 out of 20 people guess it just basically means nobody saw it. Does that make sense? Okay? That means 1/15th of a second is not enough for you to see 4 bits of information. Yeah? However, if you flash on this card for twice as long, 2/15ths of a second, then most people see it. Okay? So that means that more or less, okay, so you say 1/15th of a second, it will make you see less than 4 bits of information. However, 2/15ths of a second will make you see at least 4 bits of information. Yeah? And so therefore, yeah. How many bits can you see? It's 4 times 15 over 2, which is, yeah, 30 bits per second, at least. No? So you can see at least 30 bits per second, right? But you can at least not see more than 60. Okay? Your channel capacity is less than 60 bits, but more than 30 bits. Yeah? That's how people get it. And then when the first, and you can also tell people, say, oh, now I'm going to flash not just one of these 16. I tell you, the one I flash could only be this one or that one, okay? Then it's much easier. You just have to say, did you just show me this one or that one? Then how many bits of information do you need that to determine one or the other? 1 bit, yeah? And because it's only 1 bit, you can just show people only 1/30th of a second. Yeah? That's exactly 1/30th of a second, you see 1 bit. Okay? So therefore, these kind of experiments, when it first came out, people were really surprised. They just could not believe it. And so these kind of experiments actually repeated many times in 1950s, and then people just don't follow up anymore. So this question kind of disappeared. Yeah? And so now we need to pay attention to this. We are blind to 99% of information. That's horrible. Okay? So, yeah.
Then when one talks about to the power of 4, this is 0, 1.
Yeah.
So it means either something exists or doesn't exist.
Yeah.
And not the sheet itself has a lot of bits. It's a matrix of 10 by 10 could be.
Yeah. Because you just need to say which out of the 16 you have. And then you can use memory to recall. Let's say this is the second one of the 16. You can reconstruct this shape by your memory. You actually do not see that clearly. Yeah. Okay, good. Yeah?
Like the experiment that you described is kind of not only vision, but also kind of entangled with memory. So like.
That's right. So you show them immediately. You can say maybe it's a memory. Some people say, but you show them immediately. It's not as if, if I see you clearly, I cannot forget it in the next year. So you can test them. If you let people see it long enough, there's no memory issue. Yeah, so that's good too. So it's obviously it's a short enough exposure that's the problem. Yeah. Okay. So therefore, then question on attention, you can ask this question: how to direct attention behaviorally, neurally? That's experimental data. You have to observe it. And what are the consequences of directing attention? So I'll quickly go through them a little bit. So one is called overtly directed attention versus covertly. That's if I look at you, I'm overtly directed attention, but I can also look away, trying to kind of see out of the corner. That's overtly versus covertly. Top down versus bottom up. Top down is voluntary. I'm voluntarily trying to look at you, or bottom up. Even though I'm looking at you, suddenly the tiger jumps in, I will involuntarily look at the tiger, yeah? Because I'm trying to save my life. Okay, so then bottom up and top down. And so, for example, these are the experiments people do. They make people, this is the psychology experiment, and they're very beautiful. So imagine you make people look at this image. You say you fixate your gaze at your center, the center dot, and then your task is later on, one of these things will appear. They are all crossing the boxes, but one of them is T. Your task is to say whether this T is right side up or upside down. That's a task, yeah? And you are told that this T can appear either here or there, so 4 possibilities. And so therefore, your gaze is here. How do you know whether to pay attention here or there, there? It's difficult. But however, before this appears, if you just flash something light at this location, then people can do this better. So as if this grabs your attention over there without changing your gaze. So covertly, you just get it, like as if, okay? So imagine if you flash something on the upper right corner and then place the T right at the place of flash, they can do better than if you flash in the upper right corner and put the T somewhere else. They do worse. So this means that attention is directed by this involuntary flash, okay, involuntary by this flash. This is called valid cueing trial in psychology term. So you are just trying to cue attention of this person by, it's like I say, look. When you say look, you're pointing, yeah? Okay, well, this is not pointing. It's like a flashing. It's trying to make you look at it, yeah? Okay, and then invalid cueing trial is like, if you point somewhere but actually show them somewhere else. And you can find that, yeah, this is a bottom-up cueing because this flash is by visual input. Okay, completely, it's not your top-down intentionally you want to look that way. It's just flash grabs your attention. You can also, somebody before it shows up, it points an arrow, say, oh, it's more likely in the top left. And then you will say, okay, more likely top left, let me pay attention to that. And not there. So this is also valid. So this is called involuntary, top-down cueing of attention. So informative information tell you it could be there, okay? And this is what I mean by top-down versus bottom-up of attracting your attention, pointing your attention there, okay? And people find that if you something flash, you cannot ignore it. Even if you tell them, wherever I flash, it's actually not where the T is. They still cannot ignore it. However, these kind of arrows, you can tell them the arrow always lie to you. Don't pay attention. They can voluntarily not to pay attention to the arrow. So it looks like the involuntary part is really powerful. You cannot ignore it because it's trying to save your life. Yeah, you better not ignore when a tiger is jumping at you. So, for example, if I ask you to look for a uniquely non-horizontal bar, can you find it in this picture? You can all find it. Yeah. Did you notice there's a red bar in it? I didn't ask you to look for it, right? But somehow you all saw it. So this red bar is like grabbing your attention automatically because I didn't ask you to look for a red bar, yeah? And so this red bar is almost as grabbing your attention as strongly as this flash. So there's many different ways to grab your attention, okay? Even though you may not see the airplane engine, but this red bar, everybody sees it, nah? So you see that certain properties of an image make you see it, even though you did not see the airplane engine, yeah? Okay. There's a top-down because I asked you to look for a unique area bar. Top-down make you pay attention to that. And bottom-up will automatically grab there, doing something different. And of course, so therefore your total attention is a net result of top-down. You voluntarily want to direct your attention as well as bottom-up involuntary direct your attention, okay? And imagine if this is a vertical bar, it will actually have some bottom-up attraction to you as well because it's so distinctive, okay, more stronger, okay? You can also make it less red, then it will not involuntarily attract you as much. So therefore, there is something called saliency of an image feature that grabs your attention automatically or not, okay? What if the top-down thing that you need to pay attention to happen to also automatically grab your attention? Then your life is very easy, okay? But if the top-down thing you want to pay attention to, I see where is the airplane engine, it doesn't automatically grab your attention. Then you have to use your top-down knowledge. The engine is usually on the sideway of the plane and look for it, yeah? And so your attention is actually directed by both ways. And it can be space-based, object-based. I'm not going into detail. And then you can say, what is the hardware that does it? Remember this monkey's brain opened up like a world map, and you're trying to find out how did you move your eyes? Because attention, obviously, mostly by moving the eyes. So you chase after your eyes and say, what is the eye muscle? And then which neuron going to that eye muscle? It's like trying to trace it. And people then find there's something called superior colliculus that actually are the neurons responsible for moving your eyes, okay? And then you can say, which brain area is telling the superior colliculus to do that? Let me give you some hint. And so people find that, oh, frontal eye field is also projecting to superior colliculus with long neural wiring. And they also find this area called lateral interparietal cortex doing that. Okay, they also find V1 does that. Okay, so therefore there's a lot of suspects. Maybe these suspects trying to guide your attention or not. Maybe some are top down, maybe bottom up. So these biological hardware can give you some hint. Okay. And the retina, you notice the retina also have direct cable going to the superior colliculus. Turns out that retina alone doesn't work. You can do experiment. By cutting V1 away, then this monkey cannot shift the gaze anymore, even though the retina also has direct cable to superior colliculus. So you can always do these kind of a intervention trying to figure out which one is important, which one is not important. And then, of course, what are the consequences of directing attention? We already just said that if the attention directed there, you will do things, recognizes better than other ways. Okay, you will respond faster. So for example, this may take you longer to say what it is, and you will say it incorrectly, more likely, compared to this situation. And you can measure. So these are measurable things. You don't want to just hand waving. So experiment, you can measure. RT means reaction time, okay? So you can say, how long does a person to tell you, yeah, I found it, to report this T is right way up or upside down. This is called reaction time. Reaction time shorter, accuracy higher, then they say it more correctly. Accuracy is how correctly they say it, okay? And compared to this, reaction time longer, accuracy lower. So this is a kind of consequence of your directing attention, okay? And you can also look at what is the consequence on neural responses. You find that neurons in various areas become more sensitive and sharper feature tuning and all these kind of things. So experimentally, there's lots of these things. Then if you're a theorist, you say, so messy, difficult. Biology is always messy. But anyway, well, it's not just biology. It's also physics. Before you have a theory, it always looks messy, okay? So it's only after you have a theory, you realize, oh, it's everything very organized. So anyway, here's the messy situation. We find lots of data. Okay, and so therefore, now we need a theory. And as I said, we try to choose a theory that not only will tell us about neurons, but also tell us behavior, such as how the eye moves, whether you recognize something. Otherwise, it's just not enough. So that means we not only want to look at these things behaviorally, we also want to neurally. Which one is that? Turns out that there's no theory could do it except for one theory, bottom-up attention. Okay? So therefore, today, since this is for physics, I'm only going to talk about a theory which actually has both aspects. Okay? Bottom-up attention. Okay? So, which is actually the V1 saliency hypothesis. How lucky we are because we're having problem at V1, efficient coding, maybe it's doing something else. So in fact, when the efficient coding is no longer working so well in V1, it almost forces us to think what is something else. So it just so happened that attention needs somebody to do it, and maybe V1 could do it, but it may be wrong. Yeah, okay. Bottom-up is automatically, involuntarily make you direct your attention. Like this is flashing. If something flashes, you involuntarily will look. Okay, or at least, yeah. That means it's by external, exogenous. Top-down is endogenous. You are voluntary. You think, oh, I need to listen to Zhaoping because she's giving a lecture. Maybe I want to pass the exam. That's a voluntary directing of attention. Yeah. But even when you talk with me, if a fly, you will, involuntarily make you move your eye to the fly in the corner of your eyes. That's involuntary and bottom-up. That means it doesn't come from your, internal drive. It comes from bottom-up physical stimulus from outside world. And they also call it exogenous, because it comes from outside world to draw your attention. Good, thank you for me to, these.
What is top-down?
Top-down is that you think you are having a goal, such as, oh, I need to find a bar that's uniquely non-horizontal. So you have a reason to do something, yeah? Because that's your task, or because you are trying to look for your food to hunt, and so on. And you have a task. I'm looking for hunting an animal, I'm looking for mushrooms. That's a voluntary directing of attention. Maybe mushroom looks red, I want to look at red things, yeah. This is voluntary, yeah.
So this starts from the brain and goes out into the...
Correct, so it's called endogenous, starts from your brain. The reason I say bottom up, top down is as if this is bottom, this is top, yeah? So bottom is coming from the eye, the input comes in and drives you to do it. Top down is the top of the brain, make you do it. So that's what so they're talking about like neural network, bottom layer, top layer make you do it, or bottom layer make you do it. Okay, but another term is voluntary versus involuntary. Or exogenous, that means outside force make you do it, versus endogenous, the internal force make you do it. Thank you, okay. And so, so let's look at, let me just go say it's a V1 saliency hypothesis satisfy this, so let's do that. Okay, V1 saliency hypothesis also is very nice. You can see that selection is such an important act because remember we have to do chicken without an egg, so obviously it's a very difficult task. And so therefore you can imagine the most expensive part of the brain need to do it. Okay, which one is the most expensive part of the brain? The smart part of the brain. Normally you think the smarter part of the brain is here in the front, right? We are smarter than monkeys. Monkeys do not have as many front. And so people used to think that this part of the brain is smarter. So they always think directing attention is by this front part of the brain, not the back part of the brain. Okay? But you can also say which part of the brain having, this is a huge part of V1. Why is it 100 times bigger than retina? It must be doing something important. Okay, so this can give it a hint that it's the largest visual cortical area. It is actually very expensive. It's sucking a lot of blood. Maybe if it's expensive, let's make it do important things, which is voluntary directing of attention to save your life, okay? Because many animals also have to direct attention. They don't have this big front. They have to be involuntary direct attention, whether it's a fish, a mouse, and a cat. They may not be as smart as a PhD. They still have to, direct attention. So, okay, so now let's look at the V1 saliency hypothesis. And this hypothesis, it says, okay, something is salient. Okay, so this flash is salient. It grabs your attention automatically. You cannot ignore it, even if I tell you wherever its flash is unrelated to where I'm going to put a T, you cannot ignore it. That's the experimental fact. But, okay, so therefore let's define saliency. Define saliency as the degree of a spatial location to attract attention in a bottom-up manner, in a stimulus-driven manner, okay? So you say, okay, maybe this location has saliency 10, this location has saliency 1, this 1, so this is the highest. You can give it a scalar number, okay? So how much you're going to direct your attention to that. And then you find that I don't have to flash something there. I can also make you direct attention to that by putting 3 green rectangle here and put a red rectangle. It will also automatically grab your attention. Remember, there's a red bar automatically grab your attention, yeah? So it works as well. And so therefore, you can say this location is also very salient. Maybe not as salient as that location, but it's still salient, okay? You can measure. You can even measure, say, how often it makes you go there, how fast make your gaze go there. You can actually quantify it. You can also do it by putting all these other locations in the horizontal bar and put one vertical bar there. So it's distinctive. It will also grab your attention automatically. So flash is only one way of grabbing your attention. And so therefore, what is saliency? The saliency does not depend whether it's a red color or vertical bar or it's a flash. So therefore, saliency is a generic quantity. So then people think about which part of the brain is doing that. So the part of the brain must be not tuned to exactly what orientation it is, because it could be this is only horizontal, but rest is vertical. It works too, right? So therefore, it's not tuned to exactly what is the orientation. And it could be this is green, the rest is red, will also work. So it does not depend on whether it's which color, and does not depend on whether it's flash. So which part of brain? So people say the brain somehow built a saliency map with a high value here and a low value everywhere else. And this is a psychological quantity, which is make you behave and go there. But where is the part of the brain does that? People say, well, V1 saliency hypothesis says V1 does that. That's just a hypothesis, okay? And then you have to say, does it predict something? Can I test it? But let's also, before we do that, we're trying to look at a traditional idea. The traditional idea says, well, saliency must be not dependent on whether it's exactly red or vertical or flash. So therefore, it must be that when a visual input comes in, you first of all analyze it to what color it is and what orientation it is, what motion and depth, all these, how scale big, and then you somehow combine it into a master saliency map, where it just tell you this is 10 degrees of saliency, this is only 1 degree of saliency. Yeah? Because it's called, the idea is saliency is independent of your features. And then because of this, people think that it cannot be in V1 because V1 has all these neurons tuned to orientation, tuned to color, all these kind of things. Can't be there. It's not saliency, okay? And so sometimes theoretically we can get misled by these kind of assumption. Yeah? So therefore, people used to think that saliency must be in this higher brain area with a frontal smarter part. That's a little bit biased. We think it's very smart, it's a very difficult thing. And these frontal areas, indeed, neurons are not tuned to exactly which orientation, which color. All it needs is a neuron has a spatial map of the visual world, and some neuron here fires a lot, other neuron here fire less. So that's a traditional view. So how do we break out of traditional view to this? I will show you, okay? So then you say, which view is more correct? Traditional view thinks somewhere called frontal eye field. Remember, frontal eye field also direct neuron projection to superior correctors. So it's a very good suspect. And so people can, frontal eye field might be one of them. And so you can say, these kind of framework will give you some theoretical prediction. That framework will give you different, hopefully different predictions, okay? Only when you different prediction, then you can do experimental tests to see whether this agrees with data or that agrees with data. And we will show you how to do that, okay? So more precisely, the idea is behaviorally, you somehow in the brain, the saliency map is built. And in your V1, there's different neurons firing. This neuron firing a lot, let's say 100 spikes per second. This neuron firing 1 spike per second. And so therefore, V1 has this retinotopic firing rate map. Okay, and then at each location, Rx is the highest V1 response to a visual location X. Well, actually, in this location, there's a neuron tuned to vertical, another neuron tuned to horizontal, another neuron tuned to red, and all these kind of different neurons. But let's say there's 1,000 neuron cover, recidiv field cover this. You just choose their highest firing. Whichever one highest most, that will be the highest response to this location. And this is the bit for exogenous visual selection at visual location X, okay? So that means you say, oh, if I show this, people, 90% of the time their gaze go there and other places not. So you can have a measure of how likely their gaze go there. So the V1 saliency hypothesis is just say, well, here is a behavioral quantity, how your gaze will go. And this is a neuroscience property, how much will your neuron fire. And you put an equation. That's it, very simple, okay? That equation is the hypothesis. So you can have very simple hypothesis in brain science, even though things can be very complex, okay? So let's see how far it goes for us. So that means even though V1 neuron, so for instance, you say, what is the V1 neuron there that's firing that much? Could be a neuron to vertical, horizontal. I say, it doesn't matter. Neural activity as universal currency to bid for visual attention, and the recidiv field of the most active V1 neuron is selected to drive your muscle to move there. That's it. Okay, so you have 100 million neurons. They're all firing away. You can look at these 100 million neurons firing rate, find the neuron that fires most, go there. Okay, who cares whether the neuron is vertically tuned or horizontally? Don't mind, okay? And so that's the idea. So this is how we can think out of a box. So just like thinking out of a box like this, okay? Who cares whether you're from Iran, I'm from Germany, you're from, Pakistan. All it matters whether you have the money. You have the money, you can buy ice cream. Yeah. So here, the money is the firing rate, and the ice cream is the gaze shift. Okay. So it doesn't matter you are tuned to this orientation, that orientation. The firing rate can be used for saliency, for driving gaze. That's all we need. So therefore, this traditional framework to think that you have to go to a brain area when you are not tuned to features is a bit of an overkill. It's not necessary. So sometimes the simplest idea is better. Why do we have to wait all the way go over there to get a saliency? If I can do it right there, it's Occam's razor simpler. Okay? So that was you can think outside the box by saying that, well, it doesn't matter whether you're man, woman, which nationality, old or young. It doesn't even matter where you get the money. Maybe you get the money from your parent, you earn yourself. You have the money, and that's superior critics is saying that I'm selling you something great. It's not quite ice cream, but I'm selling you who gets my eye movement, who gets my attention. So whoever pays more gets it. Okay, that's how we can think outside the box. Okay, and as I mentioned, it could be that at this location, a neuron that's firing a lot is a vertical tuned neuron, or a red tuned neuron, or a neuron that's moving. Okay, if you walk in the forest, everything is static, and suddenly some bird moves, your attention will automatically go. So these are all salient things, and you may wonder. Okay, so different neurons sitting on the seat, and whoever fires most give this thing. This is the idea. Okay, I have a auctioneer auctioning attention. No discrimination between your feature preferences, only spikes count. This is the same idea. So, for example, if your input has one uniquely tilted, one uniquely oriented, and one uniquely moving, and so the V1 neuron tuned to one saying, oh, I pay 1 spike of money, you pay 3 spikes of money, you pay 2 spikes of money, and this auctioneer is your superior character and say, whoever pays me most. In fact, all I care is counting your money. I don't care whether you're men and women kind of thing. That's all I need. Okay, it's a very simple idea. And so, then the question is, how can you have a neuron tuned to vertical orientation respond higher compared to horizontal neurons? Yeah, is that because vertical neurons, remember we have this gain value G. Is that because your vertical neuron has a larger G? No, because you can also have this vertical bar become horizontal, everybody else become vertical. It still works. So it's not because this neuron has larger G, but because if you look at V1 circuit, okay, they make different neurons talk to each other. Okay, so let's zoom in on the circuit. This is about 500 microns, so it's about 3 millimeter, 2 millimeter. And this circuit is such that biologists have to have already stained them to make if this location is stained blue, that means most of these neurons tune to vertical. So neurons tune to vertical cluster to each other such that you can stain it blue, yeah? And if it's red, then it's tuned to horizontal. And so you can see that V1, at each location, some location has more vertical neurons, some location has more horizontal neurons, some location have these kind of orientation neurons. And on top of that, you see this black thing. This black thing is actually a cell body sitting in the vertical, in this blue patch. That means the cell body is sitting in the vertical tuned neurons, yeah? And it's sending axon neural fiber connections to other blue patches. Okay, what is it trying to do? It's trying to talk to other neurons also tuned to vertical, okay? And but it doesn't reach that far, okay? And it only reach about, a short distance. And so for people who are familiar with Ising model, these kind of things, you can say, yeah, this is like an Ising model with short-range interactions. You can think of that way. But actually, Ising model doesn't work, but you can think about this is a condensed matter physics where you have these, spin interactions in only short distance, okay? And this is the idea. It's wherever it projects to, and it does not project to anywhere that's tuned to horizontal. So this was the experimental facts. People find that what are they trying to do? neurons tuned to same orientation are actually trying to suppress each other. They have a recurrent interaction. It's a recurrent neural network. And so therefore, this horizontal bar is exciting one V1 neuron tuned to horizontal, and this horizontal bar is exciting another V1 neuron tuned to horizontal, and they are connected by these kind of lateral connections, short range enough that they suppress each other, okay? And therefore, if you have one single vertical bar, it's acting a V1 neuron tuned to vertical, but there's no other vertical bars around, so therefore, all the V1 neurons in these areas tuned to vertical are not being activated, so this neuron is not being suppressed, okay? And so therefore, this is the only neuron not being suppressed. That's how it works, yeah?
So like in the previous section when you were talking about encoding,
Yeah.
you talked about contrast and how like different quantities, when you understand the difference between them, like you can kind of...
That's right. Here, they don't have a feed-forward contrast. So you can say like this. Previously, we have like O is equals to, receptive field projection from your stimulus. Let's forget about the noise, yeah? And then you can say, okay, it's actually, feed-forward receptor connection. But in fact, there are interactions between neurons. Let's give it MF. Okay, so therefore, OI is not only getting KIJ-SJ, but also MIJ-OJ. Okay, so this is something that was not in the efficient coding bit. Except this MIJ is only between neighboring neurons. As I said, that this does not stretch that far. Okay? And so you can make it binary, then it's an easy model, but if it's not binary, it's not. Yeah. So analog neurons. And so such kind of a called like-to-like suppression is like if you neurons, they're all identity horizontal, they like to suppress each other. It's like, okay, positive charge suppress each other. Negative charge, positive, don't suppress each other. So you can say it's like-to-like suppression, such that if you stick an electrode trying to measure from this neuron tuned to this vertical, you keep sticking electrode there, but all these horizontal bars are outside the recipient of this neuron, and you turn them vertical. Suddenly, it's suppressed. Okay? So that is the experimental data. Same neuron, suppressed. And Huber and Weasel, originally when we're trying to look at these, one neuron at a time, you say, well, how about in your screen, you just don't put any of the bar, just put that bar. Then it's not suppressed, it's respond very highly, okay? And so this is called contextual inference. It's called intracortical interconnection, which is this connection. It's trying to make neurons interact with each other such that iso-feature suppression, that means if you tune to the same feature, you suppress each other. That's all it's needed, okay? And it was actually discovered in the 1970s. Remember, Huber Weasel found all these receptive fields in 1960. So in 1970, when they discovered this, it was seen as a messy biology. Why? You measure from this neuron, sometimes it fires a lot, sometimes it fires less, sometimes it fires a lot. Why? Oh, biology is always messy. You don't expect anything less, no? And so therefore, this is seen as a nuisance kind of thing and trying to ignore. Maybe in the future we'll understand. And now the future has come, we say, well, actually, it's not nuisance. It's made to make signal saliency. So if you have a blank world as one bar, that's the only most salient location, it will make you look at it. But if this were also salient, this bar is not salient, isn't it? So you can see this is signaling saliency, right? So experimentally, that indeed is the case. And this is iso orientation suppression. That means neuron tuned to same orientation suppress each other. Okay? So you can say there is a fuse there, and the context outside. The contextual input will suppress this. This context will make this neuron suppress. That context does not make this neuron suppress. Empty context does not suppress. That's it. Okay? That's how it goes. It turns out that you not only have neuron tuned to orientation suppress each other, neuron tuned to color also suppress each other, such that neurons tuned to blue color will suppress each other, and neuron tuned to red color also suppress each other. So if you are unique red, you also don't get suppressed. Very easy. And neurons tuned to, moving to the right suppress each other. Neurons not tuned to the, so therefore that's it. So therefore, all works. Okay, this is called iso-motion direction suppression. And these are 3 different neurons, but they signal at the same visual field location. Okay? And so therefore, you can imagine, well, this V1 network can explain a lot of these salient situations. Is it really true? Yeah.
In other words, can we say that selection process is an averaging process for the brain, and saliency happens when one particular area differs a lot from its neighbors?
That's right. Yeah, that's right. And somehow build this recurrent connection such that to make this happen. Very simple. It's a recurrent network. Okay. So, and then you can say, okay, let's try to test it with you can even measure. So for instance, this vertical bar is very salient in the context of horizontal bar. The same vertical bar is not as salient in the context of slightly tilted bar. So you can even make people do a visual search task. This is the psychology experiment people often do. Look for uniquely on the bar, and let's say people immediately, when you find it, you immediately press a button. Okay? And people can press button maybe half a second here, maybe 1 second there, because it takes longer. Yeah? And then using this reaction time of pressing button as a measure of the shorter reaction time means higher saliency. Okay, so you can actually quantify it by measurement, and you can also say, look for a uniquely tilted bar, but nevertheless, they somehow get distracted by it. So you can also measure by how much distraction it is. Yeah? So therefore, you can say, okay, compared to this, let's say this takes 0.8 second to do, and this takes 1.5 second to do. It must be because this is distracting you. And of course, this distraction is another measure of saliency and how fast it. So it's a little bit like, okay, one way of measuring something is by its weight, another way of measuring something by its volume, but they are all measuring its saliency. In the sense it's monotonic. Okay, the more something is, the more salient it is. And with that, you can do experiment, and you can compare across. Yeah? That's the idea. Okay? And then, so now you say, wonderful. Unique orientation should pop out. Pop out is a psychological term, means attract your attention. Unique color to pop out to attract your attention, and unique motion will pop out. And this is not very satisfying, okay? Because before this theory arrived, you already knew that was the case. And after this theory arrived, you say, now I can explain why. But what if they say, I don't care about why? You didn't tell me anything new, right? What can we do? So the experimentalists want you to tell them something new they didn't know. That's called prediction. You not only explain why of the past data, you say, well, I will tell you something you did not know, then you can discover it. Okay? And then they go trying to discover it, they find it's not there, then your theory is falsified. Okay? So the idea is because people do these visual search experiments, they have been doing it since 1950 and 60. So a lot of these things are already known. So it's very difficult for you to find something, there's something attract attention, not attract attention that they haven't done. And so this we can try to overcome. You say, why haven't they done it? It must be counterintuitive. So you need to find something that they don't have any reason to do that experiment. Okay, so you have isofeature suppression. The feature can be orientation, color, or motion. Okay, orientation, color, or motion. Is there another feature you can do it? It would be nice, there's another feature, you can do it, and only V1 is tuned to that feature. Then you get it. And people never did it. Why? They never thought it's worth doing. They didn't think it would attract your attention. That's how, just like you did not think you were blind if you were actually blind until your mother tells you, yeah? So that's the idea. It's counterintuitive, okay? So what is that unique feature? Beta V1 neuron is tuned to it, and people have never done it. And you need to predict. And we have learned it. Remember, we say some neurons are tuned to the left eye input, some neurons tuned to the right eye. V1 is tuned to it. And turns out that V2 is not tuned to it, okay? So the idea is if you put lots of bars to the left eye and put one bar to the right eye, you combine together, look like that. Okay, this bar looks exactly the same as every other bar. Now, it has a unique feature, okay? The unique feature is which eye sees that bar. And V1 has isofeature suppression. That means it will also get this location higher firing. Okay? If that's the case, if the V1 saliency happens to be correct, you will automatically look at it. Okay? Then you feel that's very strange, yeah? I don't see the difference between this bar and that bar. That's why people never done it. Okay? So therefore, you kind of have this prediction. You say, this is such a ridiculous prediction, such that people haven't done it. Of course, if it's so ridiculous, your theory is probably wrong too, yeah? So, but nevertheless, it's better to find out your theory is wrong than not find out. So therefore, you can do the experiment, trying to say, okay, this bar is only in one eye, not in the other eye, and I predict it will attract your gaze. Okay? And why is a good thing to test V1 saliency hypothesis is because V2 and higher brain areas, all neurons are binocular. Okay? Because it's all binocular, so when a neuron is firing, it could not tell you it's because the left eye input make me fire or right eye input make me fire. And this is why people cannot tell whether this bar looks any different. This is because decoding is after selection. Okay? After selection, they cannot tell. So V2 and above, they are after selections decoding. At decoding, if they don't have that information, if they don't have any neuron tuned to which eye, they could not tell. So therefore, you can make human do this experiment. You ask them, tell me if there's any bars in a different eye. They cannot tell you. They will completely random guess. So it is true, behaviorally, people cannot tell. You can do that yourself, yeah? You can say, I'm going to look at this, look at to my right eye, it looks like that. Very good. Look to my left eye, it will still look like that? Would it? Not because your left eye is moved a little, so it's a different image, yeah? But if you move your left eye to exactly the location where your right eye is, it look exactly the same. It will not look different, right? Unless if your left eye is more blind than your right eye, then you might see the difference. For most people, you cannot tell the difference, okay? So therefore, you predict something ridiculous that it's perceptually non-distinctive but will attract your attention. And then you can say, oh, let me have people to do a visual search task and see how fast they press button. People cannot do this task because they cannot tell where the target is. So it's very difficult to do this experiment. So you have to think of something else. Okay, yeah, this is trying to say V1 has many monocular neurons, so it has the eye of origin information. This is the right eye, this is the left eye. Okay, so how to measure saliency of this singleton by a visual search task to search for the singleton if observers cannot tell which bar is a target? It's difficult, yeah? So we have to think about a new experiment. So what is the new experiment we do is actually like this. Rather than make them look for a unique bar to the right eye, they say, I don't know, I cannot tell which bar is the right eye. You make them look for a unique bar that's tilted differently from other bars. However, you take one of the non-target bar, put in the other eye. They don't have to look for it, okay? But if it's exogenous involuntary attraction, they will look at it anyway, okay? So the idea is you have two locations that will excite your V1 neuron a lot. One is because it has a unique orientation, it escapes ISA orientation suppression. And another because it has a unique eye of origin, it escapes the ISA eye of origin suppression. And so therefore, your saliency map has two shouting neurons say, hey, look at me, and then you say which eye they will, which location will look to is depending on whether this fires more or that fires more, okay? And if this fires more, they will look there first. And interestingly, they do. Okay, so in 75% of the trials, they will look at it. And what happens is that when we look at it, oh, that's not their target, they immediately find a target, they press button. Okay? And then after they do the experiment, you can even talk to the observer. You say, I ask you to look for your uniquely only bar target as quickly as possible, press a button, and don't get distracted. But you did get distracted. And these observers usually say, no, I follow your instruction. I never get distracted as soon as I press a button. That means they were not even aware they moved their eye. Such involuntary nature. Yeah? And then you show them, they actually, because I have some eye tracking device and show them, they say, did I do that? Okay, now after they are told, you next time do it again, they say, oh yeah, indeed, I seem to have done it, yeah? And this is the reason why, when you actually move your eye 180 times per minute, but usually you're only aware 5 times or 10 times. A lot of the eye movement you actually do not know, they are involuntary, yeah? And so this eye movement is involuntary, okay? And so when this result came out, people find that this is so ridiculous, that can't be true. which is actually confirming that we have a counterintuitive idea about it. And because it's such significant finding, we better find out it is true or not. So therefore, multiple labs trying to repeat that. And this experiment is so easy to do. If a theoretical physicist like me can do it, then all the great psychologists can do it too. Yeah? What do you need to do this experiment? Yeah.
I actually was wondering, how do you manage to isolate one eye from the other?
You go to 3D movie theater, you wear these glasses. Yeah, and so therefore, a theoretical physicist can do it. It's almost like a thought experiment, and you can just. So all you need to do is buy a computer screen or some kind of screen where you can, whether it's 3D. A lot of gamers doing these 3D things. Then you have the glasses. That's all you need to do. So therefore, people can repeat this experiment literally within a day. And since 75% of trials they do that, you only need them to do 10 times. It's already statistically significant. Yeah. And you can, of course, try to make your experiment more exciting. Not only you can put a non-target bar in the other eye, you can also put a target bar in the other eye. They still see the same thing. They can't tell the difference, okay? Or you put every bar in the same eye and nothing eye near. They still, so as far as their perception concerns, same perception. However, in V1 saliency map, you have 2 places to attract your attention, okay? One is from a target, one is from a distractor. It's distracting you, okay? And here, this target is made super salient because this is unique not only in orientation, but also in the eye of origin, so it's really strong. And here, of course, it's just unique orientation, attract your attention. And so you find that people are fastest in doing this compared to that, and this is the slowest because they attract. Yeah.
I had a question going
Yeah.
to like whether in V1 we understand that we are seeing stuff with different eyes, or like the images are mixed and then sent to.
Yeah. V1 obviously understand enough to make you do a motor movement, which is yeah. It's like I just, hit this, and I say I didn't see it. But you actually have a motor movement. This is directed by your brain. So V1 has this ability to direct an action. And the whole purpose of the brain is action. Like I grab something to eat, saccade, things like that. So this is a motor movement driven by V1.
So can you say the eye movement is from V1 and?
That's right, because it cannot come from anywhere else. Yeah, because V2 doesn't have that signal. And retina, remember, if we lesion V1 with only retina, the monkey do not move the eyes. And the superior colliculus, which where all this motor thing, all the neurons are binocular, also doesn't have the signal. So this becomes the fingerprint of V1. It's like, you caught the criminal by the fingerprint. Okay, so it's very lucky we have this fingerprint. On the other hand, it's so counterintuitive that people have to, because it's like you need to select before you decode. Remember I say you have to have an egg without chicken. This is egg without chicken. They could not see it, but nevertheless they act to it. Yeah? It's like they saccade without seeing what it is. Yeah, so this is kind of philosophically, it is actually achieving that goal. Yeah.
I think completely understand the eye origin suppression, because I think, for example, if I see these images just by one eye, my brain still finds that saliency.
Yeah. But your brain see this. If you make observer say, just tell me if there's this input or that input or that input, they cannot tell you. They absolutely cannot see the difference. Yeah. However, they have the fastest reaction time in this situation. Okay. So the longest reaction time in this situation, you can swap the eye, doesn't matter, it still works. So it doesn't matter whether the left eye or right eye. Yeah. Just like a red flower among green flower attract your attention, so is green flower among red flower attract attention. It doesn't really matter. So it's as if V1 can see the color of that flower, but your V2 cannot. Okay, so imagine that this is as if V1, and you try to make it look for a unique area bar, but that's distracting, make you look. But only V1 can see that color. Your V2 and above are colorblind. Does that make sense? So this is indeed a very counterintuitive situation. Yeah. And sometimes this unique color actually guides you to your target you're searching for. Sometimes this unique color is distract you from the target you're searching for. Sometimes there's no distract. Okay? And however, if you make people do another task, you say, now everything is oriented the same way. You don't have to look for a uniquely oriented bar. You just have to tell me whether any of these bars is in a different eye or they are from the same eye. Okay, in this case, one bar is a different eye. In this case, one bar is a different eye. In this case, the same eye. And you don't even have to tell me which bar. And so to make the task as simple as possible, humans still cannot do it. They completely random guess. So their guess is as random as complete throwing a dice. Okay? Yeah. This is as if, let's just look at a color analogy again. You can even make left eye see 4 things, but one thing is weaker. The right eye see 4 things, but one thing is stronger. But you add together, it's exactly the same. But nevertheless, this one is unique. Okay, it will make you attract attention. But let's look at analogy. Okay, the red image is all red, a little bit less. A green image all green, a little bit more. You add together, this is a unique color, right? And if you want to find a unique color, people can find it immediately. So color is visible, and this is not visible, but nevertheless they shift their gaze. Yeah, this is another analogy to understand. Yeah, okay. And then compare with traditional model, they never predict this because they did not. This is so counterintuitive. They did not even include this which eye into this map. So this you can say is a falsifiable prediction that distinguishes theory because it's non-trivial. Yeah, other theories do not predict it. For example, you can even ask people to look for a T letter, T among L. It turns out that people take a long time to find it, normally. Okay? The time it takes to find a T grows with the number of L. So basically, they each look. Oh, found it. Okay? So if you make twice as many L's, they will take longer. However, you can make it fast by putting the T in the different eyes. And so my observers say, gee, sometimes I immediately find it. Why is that? I don't know. That's how they immediately find it. In some other cases, I keep on, because in some other cases, I put a T there. Yeah, so they keep on looking. Why is some images take a long time? They just don't understand. Yeah, so that's how it appears. Yeah, and then you may say, what is the whole point for the visual system to do this, to make something stand out just because they are found a different eye? Does it have any ecological significance? Well, it does, okay? So imagine you are looking at some kind of a visual scene. There is a surface in front of another surface. You better realize this surface is different from another surface. Okay, this is called image segmentation, yeah? So like I look at you, I better segment the part of image for you different from others, otherwise I cannot see you. Yeah? This is one of the notoriously difficult problems in computer vision. However, if you have these kind of mechanisms, you see that some part of the image is not visible to one eye. Yeah? So therefore, these are the locations where you have a contrast between your two eyes. That will make it really salient. So your attention immediately will grab to this. So if a tiger trying to hide, you can, from depths, you can get it. So therefore, it does have this ecological significance for you, yeah? So, yeah, these are monocular regions, and it will separate your figure from ground. So the figure-ground segmentation for your initial separating one object from another object to recognize something, okay? And yeah, so therefore, selection is looking, okay? We have already seen that if you look at this letter T, you cannot see that letter E, no? Hard, no? Unless you shift your gaze. You stick it there, you cannot see. Not because your retina did not sample it. Your retina did sample it, okay? I can prove to you your retina sample. So let's do an experiment to prove to you. You look at this. Okay, so I'm going to take it out. And so this E is alone by itself and still at that location. And now you're trying to look. You look at that T, you can see that E, right? So your retina doesn't have that information, yeah? Somehow when it's crowded by other letters, you just have this information button that it could not get through. But when it's in an empty space, even if you fixate on the T, you can still see it. So there you can see a lot of this is why, if I look at you, I cannot recognize your face. But of course I know you because I saw you earlier. But if I never know you, I have to shift my gaze to see you, yeah. And so we select by shifting gaze, okay? And then after we shift the gaze, then you decode. You say, oh, I don't see anything special. It's just a bar, looks like everything else. It's not what I'm looking for. Then you shift your gaze again. You find your target, okay? So that's how it goes. This shift gaze is looking without seeing. It's like selection before decoding. It's having your egg before the chicken. And that is how illogical our brain is, but we have to do it. Okay, so this is a particular theory, and maybe today we are almost there. I was trying to do something else, but maybe we should just stop and next lecture for continue. So let me give you an overview. So we have a V1 saliency hypothesis. The biggest challenge is its non-intuitiveness. We have to ask what the question is. We have to say how to define saliency, and so on and so forth. And and we have tested theory by a signature experiment that finger points V1. That's great. So this is a test by an experiment that we call psychophysics or behavior or psychology experiment. And the next lecture, I'm going to talk about these three things. One is you say, wow, you only test this in psychology behavior. Why don't you go to the V1 and have a real look? That would be nice, okay? Actually go to V1 and find that indeed this V1 neuron fires a lot and the eye goes there. And this only later we have a chance to do. So take a while to kind of get this chance to do the experiment. So before we have the chance, we actually use a software V1 to imitate. It's like you have a numerical method, yeah? To find an element in a numerical method. So therefore, the next step is we actually do a kind of imitation V1, but make sure this is actually good imitation. Otherwise, you can have your wishful thinking, build something that's not quite true, yeah? And then trying to do something much more non-trivial than these kind of stuff, okay? And then we will have some other psychophysics tests. I don't think we will have time to talk about all of them, but one of them is very interesting, because this one gives you a quantitative zero parameter prediction, okay? So the idea is to actually write on an equation without any free parameters. So the equation it's going to write is about reaction time for you to find something. And so write an equation like a maximum or minimum reaction time for finding something that's called finding something A and versus finding something B, minimum reaction time, and finding something C and finding something D, okay, is equals to minimum reaction time of finding something E and finding something F and finding something G. And I'll tell you what these A, B, C, D, E, F, G are, okay? And that is an equation one can derive. Each item is experimental measure quantity. You can measure this reaction time, measure this reaction, okay? All these, there is not a single free parameter. And this is something kind of like a physicist's dream, that you're without a parameter, then you can say, I'm going to measure 6 out of 7 of these reaction time, and using this prediction, this equation to predict the 7th one, okay? And then you measure the 7th one, see if they match. You don't have a single parameter to tune. And this is one of the triumphs that this theory actually achieved that. Okay, so I'm going to share this with you. Maybe it's a bit more meaningful to the physicist. Yeah. Okay. So much for today. Okay, and we continue next Monday.
So like when we were talking about eye movement.
Okay, announcement.
I wanted
Okay, do the announcement first. Yeah.
Okay, so I have an announcement.